but it doesn't have, some of this may be reviewed for some of you folks, some of them may be not. How many people is this stuff new material? Nobody? How many of the stuff is, or how many is this still review? Okay, for most, but maybe not all of us. Okay, well, we'll get into, hopefully get some new material a little bit. All right, now, what we're going to do here is we're going to start taking a look at a few basic principles of transcription and its regulation, starting out a little bit with prokaryotes, especially with bacteria. Let's take a look at bacteria, in other words, the domain eubacteria, or just what they call bacteria now. We'll take a look at that, because a lot of the basic principles apply to eukaryotes, just far more complex and far more regulated eukaryotes. So let's take a look at a typical prokaryotic gene and some of the key sequences and a little bit how this works. Now bacteria, I'll represent these boxes as a continuous DNA sequence, but I'll represent key sequences as boxes with lines representing kind of spacers and stuff. Okay, we have the gene here, all right. We may have a second gene nearby. This sometimes happens, not always. We'll continue on over to here. Now, shortly in front of the gene, we're going to have, I'll call it P for promoter. Okay, basic structure, common structure of a bacterial genes and its regulatory sequences. They're called an operon. An operon is one or more, in this case two, genes are controlled by a single promoter and single regulatory site. What that means, of course, is that you switch on all the genes or switch them all off, because they're all controlled through the same basic effective switch. Okay, so major sequences. The promoter, a stretch of DNA, tends to be about 30 base pairs long, and it tends to be located a few tens of bases from the start of the actual gene itself. It's a DNA sequence. The promoter is what binds the polymerase enzyme. So we'll put the polymerase here. Okay, now if the polymerase binds the promoter, it'll start up and it'll march down the gene or genes and synthesize the appropriate RNAs. But the regulatory sequence is what we call the operator. The operator binds a regulatory protein. Now most of the time you're dealing with a single operator and single promoter. Not always, but most of the time you got a single thing. Okay. Now, the other thing is gene regulation here is pretty much all on or all off. It's like this light. We have 100% transcription. We got 0 transcription. Back to 100%. We need the transcription here. Okay, operators bind regulatory proteins. And they bind what we call activator or repressor proteins. Activators increase transcription or allow it to happen. Repressors block transcription. Now let's see how these things work. It turns out not all promoter sequences are the same. Promoters have certain common features. They have certain sequences within the promoter that are pretty much universal. But outside those highly conserved sequences, you're going to have a lot of variation. Now, some promoters grab onto the polymerase like super glue and hold on to them very tightly. And that's what we would call a strong promoter. Now, if you have a strong promoter, that means the polymerase binds so tightly that the vast majority of times it will be able to activate and start transcription. So strong promoters, the default situation is that transcription occurs. Now, how do we regulate strong promoters? Now, you regulate strong promoters by binding proteins at the operator that are called repressors. Repressors inhibit transcription. And the reason they do it is this. The operator and the promoter sequences overlap by a certain amount, generally. Okay, now, the repressor protein in this case binds the activator sequence really, really tightly. So it sticks like glue. In fact, it binds that sequence so tightly it's very hard to dislodge. Now, like most proteins, they are regulated. So you can switch a repressor on or off, if you wish. Okay, now the thing is, what happens? The repressor protein is physically large enough that it actually extends beyond, let's have this, we'll put R for repressor in this case. The protein actually extends quite a bit beyond the operator sequence it binds to. And it physically blocks the promoter sequence. So, with the promoter sequence blocked, or at least partially blocked, if a polymerase comes down, it can't push the repressor off. The repressor is stuck to that operator sequence like glue. So the promoter cannot, the polymerase cannot bind to the promoter. So if the polymerase doesn't bind, no transcription. So repressors work by blocking the polymerase binding. They physically block it. Okay, that's the case we see with strong promoters. But there are weak promoters. Okay, weak promoters don't bind the polymerase very well. The polymerase may well stick to a weak promoter, but the thing is, these are non-covalent binding. They're weak interaction. It doesn't take much energy for these things to fall off. If you have a weak promoter, now proteins, you see these nice static pictures in the textbooks and things like that, but proteins in reality, they're constantly in motion. They're wiggling around, jiggling around, flexing, wiggling their shapes a little bit. It's like doing a belly dance, protein belly dance. Okay, now if it's doing that, and it's really hyper, like I sometimes can be, if it does that, now imagine if this is a weak promoter, and I'm the polymerase. I bind to a weak promoter, and I'm wiggling around and jiggling around, I'm going to fall off the sucker. And I'll probably fall off of it before I can start transcription. So weak promoters bind the polymerase weakly. Transcription usually does not happen. Once in a great while you get lucky and it happens, but normally it won't. So the default here is the default in this case is transcription does not occur. Now, how do we regulate this? How do we turn transcription on? This is where the activator proteins come in. Okay, the activator protein binds to the operator, and what it does is not only does it bind to the operator, it can bind to the polymerase. It can bind quite tightly to the polymerase. So now the polymerase comes in, so we have the polymerase binds this weak promoter. It doesn't bind very tightly, but it binds quite strongly to the activator. The activator holds it in place on the promoter, just where it's supposed to be. And now it holds it down long enough that the polymerase can activate and start reading the DNA. So in this case, you could call it positive regulation. You got to turn this thing on. So, not all promoters are equal. This is true in eukaryotes too. You have strong promoters, you have weak promoters. Now, by the way, since this is flu season, a little hint. Guess what? Viral genes have the strongest promoters in the business. And that's very good for the virus, it's bad for the host. Because what happens if you have a DNA virus, its promoter is so potent, it will preferentially bind the appropriate polymerase rather than your own genes doing that. So, viral proteins are preferentially made, meaning you get sick. A lot of times, the molecular biology types, when they're doing gene splicing experiments or trying to make clinical products, what they'll do is they'll cut the gene out and they'll attach it to a viral promoter and stick that whole cassette of DNA into, bacteria or what have you. And that way, that strong promoter means you get high levels of expression of that gene, and then you get to produce all these cool biotech products and make tons of money off of it. And help sick people out too, true. But, okay. All right, mostly make tons of money. That's the name of the game in the American healthcare system. But at any rate, so strong promoters, weak promoters. Now, we see the same analogy here when you go to eukaryotes, except it's far more complex. Eukaryotic genes have promoters. In the vast majority of cases, eukaryotic genes have very weak promoters. In other words, they will not bind the polymerase without a huge amount of assistance. That's where we can get regulation, a lot of regulation. Now, second thing, they're going to be, instead of a single regulatory sequence, a single operator, in effect, in eukaryotes, you're going to have multiple different sequences that will bind numerous different activators and repressors. We call those transcription factors in eukaryotes. It's the same thing as activators or repressors, so you have a lot more of them. So you get much more complex gene regulation. Another thing, the polymerase by itself, even with a perfectly good, strong promoter, needs a lot of other proteins to assist it in various ways. We'll see that later. It forms what we sometimes call the basal complex. Polymerase and about half a dozen other proteins are required for the whole process. Without those other proteins, the polymerase simply will not bind, and much less do its job. So eukaryotes, polymerase needs a lot of help from other types of proteins. So you have a huge complex of proteins that are absolutely necessary for transcription in all cases, instead of just the polymerase. Now, by the way, in prokaryotes, just want to mention, polymerases also bind temporarily a protein called the sigma factor. The sigma factor is required for polymerase operation. It's not part of the polymerase. It binds to it around when transcription occurs. Now, the thing is, there are several different types of sigma factors, and each sigma factor will partially determine exactly what genes that polymerase will preferentially bind to. So sigma factors, different sigma factors, give some aspect of gene regulation in bacteria. I think there's like several kinds. So that's a little add-on stuff. Okay, well that's the quick and dirty about... question?
Well, sigma factors.
Yeah.
I'm thinking about you said for bacteria, but the sigma factors are for eukaryotes, right?
Well, no, not strictly, but we have these other proteins that form the basal complex. Some of them would kind of be analogous to sigma factors, except we have a whole slew of them. So the classic sigma factors are with bacterial polymerases, and it's just this one protein. There's several different versions, but when you start dealing with eukaryotes, in effect, a whole slew of different kinds of sigma factors, each doing their own little thing. Okay, so, all right, so that's what we see. Fairly simple on-off type regulation, a little bit of control through the sigma factors, and of course control through the activators and repressors. Now, once again, in eukaryotes, we're predominantly dealing with very weak promoters requiring a lot of activation of gene expression.